Welcome back to an episode of SciAnts Streams. This episode, we are raising money for HIV/AIDS research in collaboration with the Fred Hutch Cancer Research Center. We also had special guest on today, Dr. Rachel Bender-Ignacio. Dr. Rachel Bender-Ignacio is a physician scientist and a leading infectious disease researcher at Fred Hutch Cancer Center, where she serves as assistant professor in the Vaccine and Infectious Disease Division. She's also principal investigator and the director of UW Positive Research, a major clinical trials affiliated site with AIDS Clinical Trial Group's ACTG, and is a driving force behind cutting-edge research of HIV/AIDS worldwide. With training background in global health, epidemiology, and internal medicine, MD and MPH both from the University of Washington, Dr. Bender-Ignacio has devoted her research career to studying the many facets of HIV, from prevention and long-lasting therapies to co-infections, like with herpes, viruses, and tuberculosis, immune system behavior, and the intersection of HIV with cancer and other comorbidities. Beyond HIV, she has led critical response efforts during global health crises, from founding and directing Fred Hutch's COVID-19 Clinical Research Center from 2020 to 2024, to shaping clinical trial design under pressure, proving she excels when the stakes are at the highest. Dr. Bender-Ignacio doesn't just do research, she embodies the mission behind it. Her work is about real people, communities affected by HIV, patients juggling co-infections or cancer, and those historically marginalized by inequity in care or access. Her dedication lies in bridging the scientific innovation with compassionate, equitable care. When you tune into this episode and you hear from Dr. Bender-Ignacio, you're hearing from one of the forefront investigators striving towards long-lasting change in global HIV/AIDS understanding, treatment, and prevention. And with that, enjoy the show. So, we already had a comment just before we get the intro about your earrings. Now, to be fully transparent, y'all, we chatted earlier today and did a little test of the camera systems. And I had a question about your earrings, but I was like, surely I'm misseeing. And then we spoke in an email, and I'm like, oh, I didn't missee at all. Would you mind telling us about your earrings?
Oh, yes. I am wearing condom earrings that I made myself.
There y'all go. It is, in fact, condom earrings. I was like, there's no way. I'm like, I can't ask. And
It is World AIDS Day, and your people who you thought might scare me off with their streaking definitely did not scare me off, because all I do all day is talk about sex and sexual health. So
perfect. I love and again, your shirt, not taking pride in research, like just the message of that too, and the inclusivity of pulling everyone into the sciences, like just I appreciate that shirt on many levels.
Thank you. Well, I'll have to show you the back, and then you can see our logos. So my research center is at the bottom, UW Positive Research, and then the Fred Hutch logo up at the top. So that's our two research centers put together. The HIV vaccine research center that's based at the Hutch, and then mine is UW Positive Research, focused on treatment and cure studies, as well as comorbidities research, so other viral infections, addiction, various things like that. Yeah.
Awesome. Rachel, if you wouldn't mind, just a brief introduction on who you are and what is your current avenue of research. And then we're going to get into a little bit of the backstory and then really dig into the science that you're working on today.
Yeah, absolutely. So I'm a physician scientist, which means that I'm an MD. I do not have a PhD. Some physician scientists have an MD and a PhD. I'm not quite that fancy. I only have a master's degree in epidemiology in addition to my MD. But I mainly do clinical trials and clinical epidemiology on HIV and associated conditions, including cancer. Also have a focus on HIV or in cancer in people living with HIV. And I take care of real people with and without HIV. I take care of people with other infections, and I study HIV in people.
And we have a quick question in the chat is do you have preferred pronouns to use?
She, her is perfectly fine.
There y'all go. So before we get into, again, the background, that already sounds like a really unique avenue of research and a really unique trajectory, right? Did you know for the longest time that it would be something that you'd be interested in both the intersection of cancer and HIV, or is this relatively new in the research journey? How does one identify? Because there's clearly a need for it, right? Like we know this is a need for this research today, but how did you identify and kind of end up at this really unique part of research?
Yeah, I mean, I think a lot of science, a lot of people's careers is serendipity, and it's hard to say that because you can't prescribe somebody's serendipity. So when folks ask me, "How do I do what you did?" and to say serendipity in some ways feels a little bit rude, but that's really how we got there. I've always been interested in HIV since before I went into medical school. It's in terms of an infection or a condition, what really draws me to it is that it has a biologic underpinning. It's a puzzle that we haven't completely solved yet, but most importantly, it impacts people, societies. It's based in religion, it's based in our conceptions about morals. That all sort of surrounds HIV in a way that really no other condition does. And so I just like the fact that the solutions to HIV is both the biology, but it's also changing structures, it's changing politics, it's having society react to HIV and the solutions for it and people who are impacted by HIV, in a different way. So HIV kind of all the way along. And then I became really interested in TB and HIV co-infection. TB being one of the largest killers of people living with HIV and one of the most common causes of infectious disease death globally. Um, and then when I ended up in my infectious disease fellowship after residency and was looking for research mentors, I thought about doing TB and HIV together. Realized I'd have to move to Kenya, which I think is a great sort of research hub at the University of Washington. But at the time, I had a 3-month-old and a husband who wasn't necessarily keen about moving overseas. And I ended up meeting a mentor who was focusing on HIV-associated cancers in Uganda. And I could do that work while traveling back and forth to Uganda. And there's really a lot of parallels between the problems that we see with HIV and cancer and HIV and TB. And so really got into it, got really excited about it. And, it started a new pathway. And now I've wandered back again. So
How difficult
serendipity.
how difficult was it just from like a new mom perspective and just being, a woman in STEM that traveling to and from Uganda and then doing research, like that seems like it's a pretty big lift, right?
Yeah, I mean, I think people say it's never an easy time to become a parent. I
Yeah.
don't know that we, choose the best time, but if somebody decides to become a parent, you do it and figure everything else out. I think probably the more challenging thing was being a first year infectious disease fellow working in theory 80 hours a week. It was often more than that. I think we've done a better job of it these days of kind of keeping the lid on it to 80 hours a week, but nursing a baby, trying to do all of that. And then I didn't travel internationally until my older daughter was, I think, about 18 months old.
So
Um,
not right away. Yeah, that's why I was like, oh, 3 months, and it's just like you immediately like have to ship off or something.
No, but I was working crazy hours, and a lot of people in medicine do, and a lot of people in science do. And so that's, it's just really challenging. And having a really supportive spouse was really, really important, still is really, really important.
So for those at home, we haven't really, I don't think we've ever had on someone who did like this intern specialization like you were doing. Is this the equivalent of, let's say, a postdoctoral research fellow if you go down the PhD route, where it's again like very, very heavy on the research, but for you it's also seeing patients? Like, what's the nuance there of that training?
Yeah, it's a little bit of both. So, the way that clinical specializations work is that they generally happen after either internal medicine residency or pediatrics residency, depending on which flavor. There's also general surgery residency and subspecialties after that. But so I did internal medicine residency, which is 3 years after medical school, and then infectious disease fellowship, which, in order to be board eligible to be a clinical infectious disease doc, is 2 years. But most folks that are doing kind of the academic postdoc component to it as well do 3 and sometimes 4. So that's sort of combining that clinical postdoctoral training that would allow you to see an infectious disease specialist, same kind of pathway to see a cardiologist or an endocrinologist. But then I did an additional research fellowship that's like considered to be a postdoc without a PhD.
That's really, really cool. And just also, like you said, the 80 hours a week plus a new kid, like it's kind of it's a Herculean effort that I feel like kind of goes understated, right? Of like how much work goes into something like this. Like you really have to love and be passionate about what you're doing to go down this route. Was there something that made you like really interested in this intersection of co-infection and because it feels like it goes kind of understated, underreported, right? Like there used to be a lot of reporting on AIDS, HIV when people were actively dying, just like not living past like the 30s, right? And now all of a
Mhm.
sudden it seems a little bit almost swept to the side, unfortunately. But the research that you're doing makes total sense. Like, of course, if you're living longer, you're gonna get sick from other things, right? So, what was it that kind of pulled you, I guess, towards this particular journey, and why? I guess you said you keep coming back to it. Like, what is it? Like, what's the puzzle that's just like so? Like, what's the big benefit for studying this?
Yeah, I mean, I think to go even broader than cancer, what we know is that in people who, well, I guess I'll take a little bit of a step back even further and just kind of ground us all in the same clinical knowledge of what's going on with treating folks with HIV right now. So HIV right now is treatable with one pill once a day. We also have long-acting injectable treatments that some, but not a lot of people can access right now, which could be an injection every 2 months, and moving towards longer injections. But HIV and the sort of, I would say the typical manifestations of what can become AIDS is completely preventable, completely controllable with one pill once a day, or again, sort of in special cases, these longer acting injectable medications. So it's very much a treatable condition. But what that means is that people who are accessing HIV treatment are still facing sort of inappropriate amounts of immune activation or inflammation, even despite great HIV control. And what that results in is an excess risk of cancer, an excess risk of heart disease. We kind of tend to think of people with HIV, with well-controlled HIV, as aging about 10 years faster on average. So all of the conditions that we kind of think of as we grow older happen earlier in folks, even with very well-treated HIV. And then because the immune system is in great shape, but not perfect shape, even on treatment, we still see an excess risk of infections. And in areas where there's tuberculosis, that means tuberculosis still is a leading cause of death in folks with HIV, even with well-controlled, well-treated HIV.
So I guess this take us even. I like how we're stepping a lot backwards here because there's so much context to add in here, right?
Oh my
It's so
God.
little
Yeah.
is like in the public sphere right now about this. A question from the chat is treatable and curable are different things, right? So what
Mhm.
does it mean if you are living with HIV/AIDS and it's under control, quote unquote? What does that look like in terms of patient prognosis? And I guess just even further step back, why is it so hard to cure this?
Yeah, those are really great questions. And I think part of sort of this, what we've learned about HIV, what we've learned, how far science has come and how far science still has to go. So HIV is a retrovirus, which means it starts as RNA. It doesn't have any DNA, but it works itself into our DNA and then essentially cheats and allows our own cells, or requires our own cells, that when they photocopy themselves, they're also making photocopies of the integrated DNA that it's sort of dropped into our genome, specifically in our T cells, but in some other cells in the body, mainly immune cells. And whenever those cells replicate, they replicate with HIV integrated into the genome. And whenever we transcribe our genome from DNA to RNA, we make free photocopies for the virus. Free being because the virus doesn't have its own machinery to do this necessarily. It just has a free ride in our cells, and we help it make copies. And then it has the different genes, different enzymes to be able to cut itself into pieces, or make itself into protein, cut itself into pieces, reassemble itself as a package, and then escape the cell and infect other cells. So the technology that we have, the basis of all antiretroviral therapy, or ART as we call it, is essentially impacting one of those places in the life cycle. And generally, we treat people with 2 to 3 medications because HIV is very error prone. as it's making all of these photocopies. And so we need at least 2, if not 3 medications so that we have kind of some backstops so that it can't mutate around our medications.
So the bad in this instance of its own DNA is good for it in that it can it's almost like evolution on speed. It's able to
It is,
rush through
it's
it.
exactly, it's evolution on speed. And because it can just make so many mistakes, inevitably it'll make mistakes. And the ones that work are the ones that skirt the medications that are trying to, keep their foot down on it. And so our medications right now can keep HIV from replicating itself, turning itself into an infective virion or virus, and budding and infecting other cells. But what we don't have is we have sort of glimpses into and ideas of, but not effective treatments, which can go in and either remove those chunks of DNA from our cells, or that can really prevent it from becoming part of our cells, or can get rid of cells that have HIV already in them without just killing all of the cells. And so with that kind of part of HIV, we call that the latent reservoir. So that's the part of HIV that's in DNA, in our cells, and can just be sort of sleeping there and not doing anything until our cell decides to replicate, in which case we start sort of making its photocopies for it. And so that latent reservoir is the difference between HIV treatment and cure. And this error proneness is also, and the reason, and the fact that HIV gets into the cells, the very cells that we need to protect ourselves from it, those are the reasons also why we don't yet have a successful vaccine. So if it's constantly mutating, we have different neutralizing antibodies that can neutralize HIV, but most humans who are living with HIV do make neutralizing antibodies, but not until after the HIV has sort of come past that point. So we're constantly playing catch up. So we have been able to isolate, as scientists, what are called BNAbs, or broadly neutralizing antibodies, from certain people living with HIV who can make antibodies that neutralize sort of a wide variety of different kind of mutations of HIV. But most individuals, there are some special cases of people who either by genetics or other things we don't understand can control HIV without needing medications. But the vast majority of people with HIV, when they make the right antibodies or the right immune responses, they're 2 steps behind the virus. And HIV is living in our helper T cells, which are exactly the type of cells that usually go after pathogens like HIV. So it's this sort of Trojan horse phenomenon. So there's a lot of work going on, especially at the Fred Hutch, about making vaccines. We're the head of the Global HIV Vaccine Trials Network. And there's a lot of promising immunology going on, but it's been very challenging. I mean, this is probably the most challenging pathogen we've ever faced in terms of finding a vaccine.
It's like you were saying, the immune system's 2 steps behind. And that's wild to think about, right? That you're mounting the immune response, but the thing that you're targeting is already gone, right? It's no longer what matters. And it's like, well, how do you even combat something like that, right? And then on top of it, as you were mentioning too, if you're trying to target a particular appearing protein, but it's hidden away in a cell and it's not yet active, how do you know where that is, and how do you end up targeting something like this? So it seems like it's one of those giant puzzles, and it's almost an evolutionary arms race where you have to catch up, but how do you trip someone who's running far ahead of you?
Exactly. And also, how do you figure out, so as you talked about sort of this Achilles heel also being its benefit, is that we now think that the vast majority of the latent HIV reservoir is full of what we call non-replication competent HIV, which means that a lot of it is garbage. It's non-full-length sequences. If you try to copy it, it wouldn't be able to form itself into a virus that would be capable of packaging itself up and leaving the cell and going on and infecting another cell or being transmitted to another person. And so even just sort of seeing the entire elephant at any one point in time is really hard, because we just being able to identify which are the cells that contain replication competent HIV versus that contain just sort of artifactual, remnants that it has laid down and integrated into our own genome. So it's very challenging.
And then what do the drugs do? So if you're on, right, these three different drug repertoires, what is the connection there? Like, what do those drugs end up doing that I guess slow that progression down, that allow an individual patient to have that now normal lifespan, right? Where it's not now a challenge to survive to, your old age.
Mm-hmm. Yeah, so there's a variety of different mechanisms. The newest one, the kind of hot, sexy one that was the Times, or that was, I think, the breakthrough of the year that Time Magazine put out in 2024 and kind of all throughout the world, is a medication called lenacapavir that now is proven to be almost 100% or 100% effective at preventing HIV acquisition when taken as a twice yearly shot. So this is phenomenal. That medication is called a capsid inhibitor, which works in actually 3 different places in the life cycle of HIV. It prevents it from sort of taking the capsid, which is the kind of capsule that the virus is in. It prevents it from disassembling itself so it can enter the nucleus, and it prevents itself from reassembling itself when it's trying to kind of go the other direction and make new virus, and it prevents it from being able to bud out. We have nucleoside reverse transcriptase inhibitors, which kind of they do chain termination. So they prevent that RNA from getting turned into DNA because they gum up our transcriptases, the human transcriptases that would be sort of reading the sequence and helping lay down those copies. Protease inhibitors, which prevent those sequences of proteins that get spit out from getting cut up into the pieces to turn into the right pieces of HIV to be able to reassemble themselves. So essentially what they do is prevent someone from having the ability to have cell-to-cell infection in their own body that would then harm their own immune system. And so when we stop killing other immune cells within our body, we're able to have a totally normal almost normal immune system that is capable of fending off all of the other pathogens that our T helper cells and our macrophages are generally keeping us safe from. Bacteria, fungus, other viruses. Most commonly, folks with advanced HIV who are not on medications or who get diagnosed very late have the hardest time with other viral infections and fungi and mycobacterial infections like TB, because those are intracellular pathogens, so things that are inside our cells, and we need those T helper cells to be able to attack those. And really the other amazing, phenomenal thing about HIV treatment is that because we're not making replication competent virus, we're not making whole viruses that are going out into our body and infecting cell to cell, we also don't measure the viral RNA in our blood. We're not seeing any of the viral RNA, which means that we can't transmit it to anybody else. So say this with me, the risk of transmitting HIV to someone is 0 when people are effectively taking their HIV medications. It's not almost 0. It's not probably not going to happen. It is 0. So that is what we call treatment as prevention. It's really a message that, more people need to hear, is that amazing message that HIV treatment not only protects the person who's taking that medication, but also makes it impossible, if they're taking their medication well, to pass it on to a sexual partner.
I think that's a critical thing that's misinformed, misidentified to a lot of people, that chance of being 0, right? It's always like, oh, there's a little bit of a chance, right? And the fact that it's 0, that's huge.
Mm-hmm.
And that's a big breakthrough.
Yeah, it took the US CDC a long time to say it was 0, even after the WHO said that it was 0. But it took having thousands of people and millions of people having sex with people who are serodifferent, meaning one partner has HIV, one partner doesn't have HIV, to really say that the risk was 0, 0, 0. And they did a huge study in East Africa. And essentially what they found was that there were a few transmissions of HIV. And in every single case, after studying it was like thousands of people who had sex millions of times. In every single case, either the person who was living with HIV in that partnership was not taking their medications and their virus was not suppressed, or the partner who acquired HIV's sequence didn't match the partner that they thought it was from, which means that they got HIV from outside their house, which can happen when you're in an endemic setting. You know, if you live in a country with 10% of people, 1 in 10 people are living with HIV, and one of your partners is doing a great job of taking their medications and there's somebody else, then it was very clear that in that study, 0 of the transmissions were from people whose partner was taking their HIV medication well.
And I bet that when there's kind of reporting on something like this, you're not like the popular media doesn't necessarily want to report on that side of things, but rather it's like, oh, it's not 0 necessarily, but you're actually looking at the data itself. You're identifying what's happening, and you have to identify all these variables when doing this patient data. And think that that dovetails nicely as we are now on the front page carousel, Rachel. We're on the front page of Twitch now. Um,
Nice.
could you dive a little bit into some of your research now? The difficulty, like you're kind of already touching on it, the difficulty of working with humans, right? A lot of the guests we've had on have worked on fruit flies and zebrafish and mice, right? But here you are, you're working with human patients on not just HIV, but other factors as well. What is the big research question that you're chasing right now, and how will, you know, fundraising efforts help you achieve something like this?
I mean, that's such a huge question. I want to say it's the $1 million question because it actually is. It's probably more than $1 million. It's definitely more than $1 million. But we're working on several things. So, and that's great. When I'm the director of UW Positive Research. Yes, we're fundraising for Fred Hutch. I have a joint appointment between both places. My research unit is physically located at Harborview Medical Center, which is our county hospital. It's where our very large HIV clinic is located. And so it makes sense for my research, which mainly engages people living with HIV and hepatitis and other infections to be coming to. But our partner organization is the Fred Hutch, between myself and our sister unit that studies HIV vaccines. So in that research unit, we can answer more than one question at a time because we have a variety of different clinical trials going on. I would say the three main thrusts of our research right now are one, HIV cure. So we have a number of studies looking at ways to, if not completely cure HIV, ways to sort of keep the virus either in lockdown, keep it from being able to wake back up out of that latent reservoir without taking medications. So that's one. I'll call that HIV remission, HIV cure, HIV remission. The second bucket of research would be in long-acting antiretroviral therapy. So asking people to take a pill every day for the rest of their life is very challenging, especially if we expect the life expectancy of folks with HIV to be potentially 80 years, which it is. So, and people have all sorts of challenges in life. People have, unstable housing, people who have mental health issues, people have substance use. The stigma of putting a pill in your mouth every day reminds you of the condition that you're living with. And for some folks, that's really, really hard. So for all of those reasons, we're working on longer acting HIV medications that could be injected once a month, once every 2 months, maybe once every 6 months. So we have some studies going on there, and those studies are specifically focusing on folks who are having a hard time taking their medication every day. So folks that have, structural problems with access to medical care or difficulties taking their medication. And then I'd say the third bucket is HIV-associated comorbidities. So aging, cardiovascular disease, hepatitis, other conditions that disproportionately impact people living with HIV or that happen kind of younger or more frequently in people with HIV despite having good immune control over the virus.
How do you identify said patients to help? That seems like it's a really, there's a lot of people that need this help, right? It's not a
Mhm.
small niche community by any stretch of the imagination, right? So how do you, I guess, first of all, get public buy-in? Because some of
Mhm.
these folks of marginalized people have, undergone very questionable treatments in the past, right, when it comes to any kind of medical intervention. And then how do you ensure that you're hitting all these different populations to know that if this is going to work, just in terms of the buy-in, not even yet the science of the drug?
Yeah. those are probably the most important questions, more important than the science that we do itself. So I belong to a network that's called ACTG, Advancing Global Therapeutics for HIV/AIDS and Other Infections. And that's a global network. We have a global community advisory board, and we also have a local community advisory board in Seattle. Our sister unit is the HIV vaccine trials unit. That's part of the Fred Hutch. They're part of the HIV vaccine trials network with global community buy-in, as well as a local CAB or community advisory board. And essentially, HIV research has really set the standard for community buy-in. I think a lot of other conditions now involve people being treated for those conditions, other community stakeholders, but it was really AIDS activists in the 80s and 90s who banged down the doors of the NIH and said, you're not finding us answers fast enough. You're not doing this. A lot of people weren't getting access to trials or new medications because scientists said, this is too dangerous. We're not giving it to people yet. And people who were living with or dying from HIV at that moment said, well, what about what we want? And what about what we need? So I think since that moment, most HIV researchers have been really dedicated to partnering directly with community. We always ask what people's priorities are. What should we be researching? I think that's why there's so much emphasis on aging, because we have a big community of folks who've been living with HIV since the 80s and 90s, and their HIV is treated, but they have a medical list that's like 2 pages long, and they say, okay, great, I can treat my my HIV with 1 pill once a day, but what about all this other stuff I'm enduring that my friends without HIV don't have? So so we listen to the community. We have a community advisory board meeting every month locally, and we run all protocols or potential protocols by our community advisory board for input. People tell us, this seems too invasive, or why are you doing this? Or have you thought about asking these types of questions? And then when we develop protocols, we always have a community member on every single protocol on our data safety and monitoring board. So there's at least one person from the community, someone who's either openly living with HIV or otherwise states that they're impacted by HIV, weighing into the science with as much input on that protocol as any of the scientists or biostatisticians.
What is the role of said community member, like in terms of the weighing? Because like you said, that seems really, really important to have that buy-in and to have members of said communities with a voice. How do you balance it where it's like, you're doing the science and you might see what's efficient and what might work in a patient versus also the balance of that buy-in and then just the feasibility, right? Because you were saying that not everyone can take a pill every day, right? So how does, when you're getting the feedback from one side versus the other side, how do you pull those two things together and make it into a cohesive, almost treatment plan?
Yeah, it's really challenging. I think one of my other passions is how to figure out how to do clinical trials better. And I sort of have been working on this idea in a paper that hopefully will be published soon called What If Marie Kondo Wrote a Protocol? Because what ends up happening is that we end up having 75 cooks in a kitchen.
Yep.
And, what about adding this survey to ask people, how their experience of this new product is? What if we, collect some cells to study this other thing? What if we, what if we? And you end up with this cluttered thing that's so impossible that it actually is so burdensome for the person to participate that they almost can't unless they don't have a job. And it's so burdensome for our research center to have all of those steps. That said, a lot of that kind of weight of what gets built into a protocol is for safety and for ethics. I'm glad you brought up that obviously marginalized populations who are disproportionately impacted by HIV are also those who've had, in many cases, the worst experience with science, with medical care. And so every protocol also has layer upon layer of safety and ethics reviews. And that's part of what the community member also does. They read our consent forms before they're finalized. They make sure they're understandable. They make sure that having read the protocol and the science, having read, if we're going to give somebody a medication, whether it's already approved or investigational, that were adequately reflecting the risks that they may face, adequately reflecting the benefits that they may get, the person participating may get, or saying, you might not get any benefit, but science, other people may get the benefit. And so just really weighing into all of that, making sure that we're balancing the amount of information that we want to learn from a study versus what would be reasonable to ask a person to undergo in the name of either improving their own health or just altruistically for science.
What is a reasonable thing to ask someone when doing a clinical trial? I think we don't usually get exposed to these clinical trials, right? And you're like in the dredges of it. You're doing it. You're running the experiments. You designed the drug, right? What exactly is reasonable to ask a patient? And I guess what would be your dream scenario of what you could ask for? What is reasonable? And are those differences exacerbated by different communities and different backgrounds, right? So are there some communities that you really want to help that some of the questions that you want to ask are just like this is going to be too much, and vice versa on others? Like it just seems like a logistical nightmare, and the fact that y'all can help people is huge just to begin with, right?
Yeah. So I think the kind of short answer to that I'll give first. I know I don't give short answers. The short answer is that there's not a one size fits all thing that you can ask of people. What you ask of people is sort of proportionate to what they're experiencing from their condition, as well as sort of how intense the research needs to be to answer the question. And I think what we're trying to figure out right now is sort of a scientific community a little bit is how to right size that. So we want to have the highest amount of safety controls and information learned from a study in which we're studying HIV reservoirs and trying to work for a cure. For those studies, we're usually asking people who have well-controlled HIV, who know a lot about their health condition, who really are interested in the science of it, who, in some cases have been sort of thinking about it and reading about it, sign up for doing something extremely intense that they know that they might not actually get any benefit from, but will be helping somebody in the future potentially be cured of HIV or to be able to have their immune system take care of HIV without treatment. On the flip side, when we do something like implementation science, which means taking something that we know works in the ideal perfect population and trying to see how do we get it to new populations or how do we get it out into the clinic, those studies we want to be very light touch and ask almost nothing of the person. We want them to know that they're in a study. We want them to consent to being in the study. And otherwise, we want to sort of mess with reality as little as possible. Otherwise, you have the Hawthorne effect, right? Because you're watching it, so it changes. So we really kind of vary that risk and that intensity based on the question that we're trying to answer.
Cool. So it's again a lot of that personalized medicine, but now a personalized almost clinical trial, right? Where it's like you have to be very aware of the patient that you're pulling in, what you can ask, how you can ask, and how that's going to inform your research question.
Absolutely. Absolutely. And for studies that are focused on answering a question that's specifically like for folks who are having a challenge taking their medication, we're specifically looking for populations of people who are often using substances, may have unstable housing. For those types of studies, our minimum standard is we need a way to get a hold of you, and you may not own a cell phone, but you need to have some way that you can check in with us, a case manager who you give us, permission to contact on your behalf, a place we can leave messages with. And we want that study to be as light touch as possible because we're trying to design an intervention for people who are having a hard time sticking with medical care.
So let's get into, we have a lot of questions coming in from like the drug trials perspective on its own, but let's get into a little bit of like the science behind what is the new thing that you're trying to do. So what is the new treatment's goal that you are trying to take? Like we touched a little bit on, it's like not daily taking of drug. What is the new thing trying to do? Like what is different about it?
What is different about it? So I think there's a couple of different angles on long-acting HIV treatment and prevention. I'll kind of go across the spectrum And that I think a lot of what is converging in HIV immunology and in treatment is this idea that HIV prevention and treatment are looking a lot more like each other, and HIV cure and vaccinology or HIV prevention are kind of looking a lot more like each other. So in some ways, our research is asking very similar questions in the prevention and in the cure realm. So there's sort of two buckets of making a treatment or a prevention longer acting. One could just be a new mechanism of getting it to be distributed in the body. So we've got long acting medications that everybody's familiar with, like Depo-Provera, birth control that is a shot that lasts 3 months, or a NuvaRing or something like that. I'm saying the brand names because that's what people know, and there's long acting psychiatric medications and things like that. So those are just pharmacologic innovations or sort of new ways to deliver probably the same drugs or similar types of drugs that aren't really thinking differently about how to treat or cure HIV differently. But some of the prevention and cure techniques that are sort of, again, kind of converging is new immunology, which is working on ways to either teach our immune system how to respond more effectively or give us passive immunity. So passive immunity is getting an infusion of antibodies. We learned about this a lot, I think maybe the public learned about this a lot during COVID with a lot of the COVID treatments that were these monoclonal antibody infusions, so synthetic antibodies that were used to either treat or prevent COVID. Actually, all of that science, I won't say all, but a lot of that science came out of HIV science, where we've been working to put together these broadly neutralizing antibodies for either HIV prevention or HIV treatment or cure. And sort of in this sort of cure realm, what we're seeing is that, people have different goals for what cure means for them. Some people need to test HIV negative or have HIV eradicated from their body where they never see it again. And other people just need to not take a medication every day. So long-acting treatments and HIV remission from kind of an immunologic standpoint, meaning maybe giving somebody intermittent infusions to boost their immunity or to give us passive immunity, sort of functionally do the same thing, which means that, maybe we have a future pretty soon where somebody could get a couple of infusions or a couple of injections per year and not have to take daily medication. And that can be either kind of via the classic HIV medication delivered differently or via these sort of novel immune approaches that keep the HIV under control without treatment for a longer period of time.
That's really cool. So you're going down several routes to try to get at this same answer, right? Of like repackaging existing things to last longer, right? Based on mechanisms that we already know how they work. You're just now repurposing, right? And remaking, and then novel techniques as well, and just pushing forth both. And with the end, I hope, right, everything works. But if one doesn't pan out, you got the other one that's going.
Right. And there's even been some recently completed clinical trials in which people can get maybe 1 or 2 infusions of a BNAB, one of these neutralizing antibody solutions, plus a long-acting HIV injectable medication every 6 months. And so the two of those things put together could equal that person's HIV treatment. So a little mix and match there. And the same thing for prevention. Generally, we need less medication to prevent than we do to treat. Sort of a general principle throughout medicine. But both of these kind of immunologic and long-acting medications are going into the prevention realm as well.
What phase drug trial are you working on now? And how does one progress? I think you said mentioned it was phase 2 clinical trials currently. How does something like that be progressed? So the chat was wondering, like, what does a result look like for you where you're like, okay, this is safe for X person or X number of people, and then now you can progress to a larger population size or testing it on now individuals that aren't as healthy. Like, where do you make that fine decision of what the next step is?
Yeah, so we have fewer clinical trials going right now than we do in a normal year because 2025 is not a normal scientific year, as all of us know. But we often have 15 to 20 trials open at our research center at any point in time.
Wow.
And those can actually span all the way from first in human, meaning that this is the very first human who's ever gotten this medication after it having been tested in macaques and in beagles, and then it goes into people. So we recently completed at our center a first in human study of a novel nanoparticle HIV delivery system. And then we have everything from there to phase 4, meaning it's already approved and we're looking at it in a new light, or we're looking at it for a new population. And I think the key thing is that I never make these decisions by myself. So I think all of this is team science. It's never me saying, okay, I think I'm ready to put this into phase 2. This is all very carefully thought about at each stage, from usually a data safety and monitoring board who's looking at the results in near real time to make sure that the people who are on that study are safe and that they're having effect. If that's the intention, is that to be able to find out effect in the studies. And then each phase is also approved by the FDA as well if it's a new drug. So there are many, many layers of people looking at this, starting with biostatisticians to make sure we're using exactly the right number of people in a study, because we don't want to have too small of a study where we can't answer the question, but we also don't want to enroll a single person more than we need, because that's extra risk to that person, that's extra time and money wasted. So it's a biostatistician helps us right size it, helps us make sure that we're answering the correct question and measuring the outcomes in the right way. Again, ethics folks involved, community members involved, virologists, immunologists, many ologists, cardiologists for some of our research. And then at the end of each study, our results are posted on clinicaltrials.gov, so everything is publicly available, whether or not there's a manuscript published in a journal, which we're obligated to do, but may take some time. But within 1 year of that last person exiting the trial, we're required to put the results up on a publicly available website. We have to report to the FDA. We have to report to our ethics boards. So there's a number of checks, and then generally a big group decision that says, okay, we're ready to move it to the next phase, or we're ready to create an application to the FDA for approval of a new drug, which is generally not me. It's generally at that point as a company, or it may be a university, for example.
So at which of the stages are you most involved? You mentioned that there's stages where there's macaques involved, the macaque testing,
Mm-hmm.
and then I believe phase 1 is when you're testing on healthy individuals, right, for getting
Mm-hmm.
dose responses. Which one of
Mm-hmm.
those are you involved in, or do you get to do all of those?
I get to do all of it, which is so fun. For our first in human study, it's important to look at the data from the macaques and make sure that we think that the drug is long acting enough to make sure that it's safe enough in those animals. The FDA recently, for one of these compounds, said, it looks great in macaques, but we know that beagles are more sensitive than monkeys are immunologically, and if there's going to be a reaction, we want to know about it. So let's test it in a second species, or and that's actually a third species, because most things start in mice or rats before they go on to macaques or to dogs. And then we look at the safety, we look at all of that before we look at the human data. So I don't do any of the experimentation with monkeys myself, but I want to look at that we call it NHP, non-human primate data, before we do anything with people. And then we get to do everything from the very first injection ever in a human all the way up to here's something that's approved, and we just want to see if this drug answers a different question or maybe a new use case.
And in that instance, it's something that's already gone through safety testing. You're just kind of using it for, I guess, basically to see if there's a side effect that can help someone, right? Like if there's a side effect that's now a treatment in another case. Is that a kind of a fair way to put it?
In some cases, I think that that's very common with immunologic drugs or some of these kind of more broad medications. So, for example, we recently concluded a study with a drug called nilfinavir, which is an old protease inhibitor for HIV that we don't use clinically anymore, both because it's too weak and it also has a lot of side effects. But it turns out that some of those side effects are on mitochondria, they're on our T cell cycling, they're involve kind of getting into the machinery inside of a T cell. So taking that medication in high dose for a very long time didn't work so well for HIV. But when you think about it for Kaposi sarcoma, which is what we were using it for, we can tolerate a lot more side effects for chemotherapy, right? Because if we're taking something that's sort of like, it wasn't a great HIV medication because it has too many off-target effects, but those off-target effects are way less toxic than taking cytotoxic chemotherapy. And someone's only going to take it for 8 weeks or 12 weeks, and they have a condition that currently doesn't have a great treatment, like Kaposi sarcoma. We are willing to look at that side effect profile very differently, especially when those off-target effects are sort of impeding our cellular function in a way that we think might get in the way of the tumor growth.
So I think one thing that people maybe aren't aware of is the importance of this research, not just for the direct question that you're asking, but also the side rabbit holes that you're going to be able to answer, right? It's like in basic fundamental science, studying a jellyfish protein GFP got you now a molecular tool that everyone uses that's like revolutionized the field, right?
Right. Including for HIV science.
Right. And then you as well now, so you're in like treating HIV, but you could
Mm-hmm.
find something that could help in cancer or in any one of these other diseases. Could you talk a little bit about like one of these serendipitous moments where it kind of, you're looking at one treatment and it turns out it could help in something totally different and kind of tying
Yeah.
it back to why then this is important to fund? Like you mentioned, it's been an off year for research for many of people, right? Of like, we just got an anonymous $50 donation. Like these things add up and can make a difference. I think just really highlighting that it's not just for people with HIV AIDS, but you could be helping people across the board, who've never been touched by this in any way.
Right. Right. So I would say it's maybe a little bit less common that the medication itself has off-target impacts. I would say it's really the immunology and the virology that probably impacts other diseases the most. So a couple of good examples are that in understanding when I said that, people who are taking their HIV medication well have almost normal immune systems with that kind of, asterisk. That asterisk is that people with HIV still have what we call kind of more of an exhausted phenotype to their cells, which means that their cells, some of their T cells are kind of tired from trying to fight HIV all of the time, and then may not be as effective at spitting out toxic chemicals or going around and killing other viruses or other things. And it turns out, or tumors. And so some of these molecules that people discovered in looking at this phenomenon called immunoexhaustion with HIV are checkpoint molecules. And checkpoint molecules, it turns out that when you block them by giving a checkpoint inhibitor, are some of these medications like pembrolizumab or nivolumab, which have just completely revolutionized cancer treatment for a wide variety of cancers over the last 20 years. So for a lot of medications, when people are taking, or a lot of cancers, people are now using kind of more immune therapy than sort of the standard cytotoxic chemotherapy that just blasts all of their cells. A lot of these nuances in understanding the immune system and where to actually tackle cancer came from our understanding of the immune system with HIV. I think that's true for chimeric antigen receptor T cells, so CAR T cells, gene therapy. A lot of these things are kind of, people have been really working on them for HIV. And gene therapy is, I think, working first for sickle cell disease and for other things like that. But again, a lot of this work has sort of been coming out over the decades with HIV. In terms of sort of off-target effects, what we're also seeing are things like, people that are taking Paxlovid for COVID. That medication, nirmatrelvir, doesn't last very long at all. And so you can make it last long enough to just have to take the medication twice a day by pairing it with ritonavir, which is an old school HIV medication that kind of makes our liver not process it so fast. So that's specifically an HIV medication. But really, probably, I'm only overstating it a little bit to say that the speed with which we were able to find vaccines and treatments for COVID was really, really from the foundational research that has been done in vaccinology and immunology and in virology for HIV. And importantly, I think having these big networks that are up at the ready, the HIV Vaccine Trials Network and the ECTG and others who have that infrastructure, and folks like my colleagues who know how to run clinical trials, so that when we needed to do a COVID clinical trial, we're all standing there, we're all ready, we're all trained, we all know how to do this. We just had to switch pathogens really quick. And we did that again for Mpox. So I think so many different ways that HIV science has really impacted a lot of different other conditions.
See, that's a big thing to highlight as well, because a lot of people, right, were like, "Oh, the COVID vaccine came so quickly, and how could it have come so quickly?" And you're saying like, well, not really, because the
Right.
mechanism of how to cure and treat it and develop things we've been working on for a long time, and it's just like you said, you just switch the pathogen, did the same protocol, got the answer, switch right back. And that is kind of the remarkable nature of what it is that we do in science, that it's not just for a single disease state, that you're learning fundamental components of biology that you can apply to so many other things. And that goes really,
Mhm.
really understated.
Yeah. I mean, and even just again, the idea of bringing it into a clinical trial, having sites around the world, like the HVTN, again, which is headquartered at the Fred Hutch, and the ECTG, which is now headquartered at the University of North Carolina, but has a site here in Seattle and all across the world. We have teams at the ready. We had community. We knew how to get community involvement. We knew how to get involvement from all of the different types of people, and how to just get the machine running so quickly, because people have been doing this. And our leaders of our networks who led the COVID efforts are the same people who were in the room when it happened with HIV in the early 80s, including Tony Fauci, or maybe even most importantly, Tony Fauci.
That's yeah, I really appreciate you mentioning that. And again, tying back to the importance of keeping these things funding, that it's not an isolated disease that you're treating, right? It's not just like you're funding cancer, you're funding HIV. These things are all as interdisciplinary as the science that you're doing, which is, can I just say how cool it is that you're getting data from like rhesus macaques one day, and the next day you're looking at dog data, and the next day you're looking at people data, and just linking everything together? Like that that's huge. That's an immense challenge to be able to be running on a day-to-day basis. But I bet also very rewarding to be able to see it throughout the lifespan of its development.
Yeah, it's so much fun, and it's such a great challenge. And I think one of the best things about being, I wouldn't say I'm super senior, but about being mid career or being more senior, is that I have a number of colleagues, a number of mentees who are working in areas that are more broad than I can work any day. So I work with a fantastic epidemiologist, PhD epidemiologist, who has sort of taken on all of my cancer and HIV research that I started on as a postdoc and had to put down for a couple years, and is really working. She's an epidemiologist and an implementation scientist, and has really grown that into her own, and is really leading that. And I collaborate with her, and I check in with her and help mentor her, but that's really her own. Separately, we're working on epidemiology and outcomes related to HIV with these big US data sets that have 20,000 people living with HIV, and we can look at clinical outcomes through some, pretty pure epidemiology and in that way. And I don't do all of my own analysis anymore, but I have fantastic colleagues and mentees who can do that analysis, and I can sit and brainstorm with them about how to ask the question, how to avoid bias, how do we measure something correctly. But then, ultimately, we just have this really broad interdisciplinary team across a number of countries who can kind of take on each of these pieces, because none of us have the expertise to do this on our own. And that's really what team science is.
When looking at those kind of really large data sets from patients, how difficult is it to get answers from cross data sets, right? Because everyone is so different. You can't control a person like you can a fruit fly, and not just with the genetics, but right, the temperature, humidity, diet. How do you account for all these variables that are present in people to then be
Mm-hmm.
able to make these informed decisions about these kinds of clinical trials, or just even figuring out dosages, or just any of the fundamentals?
Yeah, I think that's a great question. I mean, one of the key things is we asked completely different questions. And we also work on our data sources. So the Centers for AIDS Research is a big collaboration funded through the NIH and has sites across the country. The Fred Hutch and UW is one site, but there are 9 other sites in the country who all contribute to this data set called SciAnts, which is the Center for AIDS Research data set across these 10 big academic HIV clinics, and we all collect the data in a particular way, and it's all vetted in a way that it works all together across these 10 sites, which are very geographically representative of the United States. But even so, we don't try to answer questions of like, does this cancer therapy work? The type of questions we might answer is, how has the risk of cardiovascular disease changed over the last 10 years in people living with HIV? What is the percentage of people who are getting, hepatitis vaccines, and how protective are hepatitis vaccines? We recently worked on a study of Mpox and looked to see what the vaccine effectiveness was of the Jynneos vaccine or the Mpox vaccine in people living with HIV, because there's no clinical trial that can enroll that many people. But when you look across 20,000 people or 25,000 people in the United States living with HIV, some of whom got the vaccine, some of whom didn't, people are all behaving differently as people do across the US. So you don't know what they're doing, but you can look at vaccine effectiveness, which essentially means, of the people who were vaccinated versus not vaccinated, how many people did or didn't get Mpox, and calculate that. So it's just, we did a lot of COVID research that way. We're not trying to answer kind of therapeutic intervention questions that way so much. We're asking questions about population health more that way.
So it's like a broad question that you can kind of get at with just the sheer number of patients that you're looking at, and it's not very nitty-gritty answers. It's just patterns.
Some of them are very nitty gritty answers. I think that we have to for real card carrying epidemiologists. There are lots of ways to make sure that the answer is very specific and is very rigorously obtained. It's just, again, it's a completely different answer than one would obtain within a lab or within a clinical trial. Because the truth is that we need all of these things together to understand, how systems work. And oftentimes, the epidemiology, these sort of big data set research, can be either hypothesis generating, meaning it's helping us kind of come up with a question that we then take back to the lab or then take back to a clinical trial, or it can be kind of to confirm something. So we can say, maybe we saw this thing in a clinical trial, but the question is, does it really work in people who are behaving as people do? And that's a whole different question from, did it work when we took people who looked exactly this way and fit in exactly these boxes and we watched them very carefully, right? So we need sort of this real world data to look at trends, to come up with questions, to then take that back, into clinical trials also, and make sure that we're answering the right question for the population and not just, what seems interesting to one of us.
I'm loving learning all the nitty gritty to this because it just shows, right, how much goes into identifying what a target is that you take a next step on. And I like to, you're like, it's this like very interdisciplinary team science. Like you have all the various experts and then you have community members on board as well. Like it's just a very beautifully well done art almost. And we actually just got, I think we can seg now a little bit to the financial side of things before we get back to a little bit more on the community buy-in. We just actually had a donation for $42 coming in, which by the way, thank you all very much. That we are raising money again for HIV/AIDS research today. In collaboration with Fred Hutch Cancer Center. We have incredible guests on with us tonight as we're going through the ins and outs of clinical research, of drug development, of drug discovery, of community buy-in, of like I'm just like kind of jaw dropped on most of the conversation that we're having tonight because I'm having so much fun asking these questions. But can you just give us a perspective on like why something like a fundraiser, where a a person at home who has $5 to give to something like this, why does it make a difference? Like just a little bit of a perspective on how much something like this can cost. Because we're talking about like, macaques, dogs, rats, mice, people, getting people in, developing, timing, data sets, analysis, teams, people like this seems
Yeah.
like it could be a huge operation in terms of finance. Is that a fair assessment to it?
Yeah, absolutely. And I think, when we're talking about funding an institution like the Fred Hutch, and why I'm so thankful to belong to the Fred Hutch, is that there needs to be some degree of infrastructure that's there and operating all the time, because we can't just, for example, write a grant, get the grant, hire all the people to do the thing, the project's over, we wind back down, we wind back up. We've got tremendous amount of expertise and continuity, and the life cycle of a clinical trial or of clinical trials is oftentimes 2 decades between when it leaves non-human primates until it becomes an FDA approved drug. And oftentimes that sort of drug development commercialization piece, the reason why it's done by industry is because it is so expensive. But if we take back, it's going to kind of go back a step and think about what the Hutch is doing, is that it's often funding us to do some pilot work, to be able to just run a small number of samples, or to test something in a small number of people to get a little bit of preliminary information, to keep funding our community advisory boards, some of these things that are around the edges that grants never pay for. And the things that you need to just keep going on a daily basis. You need to have a strong institution behind you. Otherwise, again, you'd have to sort of spin up and spin down the entire infrastructure for every grant, and that would be incredibly inefficient.
And the stop-start nature to it, like you were saying as well. Like imagine there's a gap in that kind of funding. What do you do?
Yeah, and I think that we're looking at a world where, there probably will be a lot of gaps, if they haven't already happened this year. My research group has had a huge number of gaps this year, because all of our clinical trials were on pause for the majority of this year. We heard testimony in Congress that clinical trials were not impacted. I'm here to tell you that, my clinical trials were impacted. We were keeping the people who were on trial safe. We were seeing them, we were collecting their data, but we weren't enrolling any new people or opening any new studies, because with the small amount of money we had, we decided that, you go back to basics and you take care of the people who are with you right now. And I think that's possibly the future. I mean, that's always the situation of research, but I think the future of research is that it might be more spotty with having dependable funding.
Can you comment on the hardships of something like that, of like a mid pause on a clinical trial? Because naively, right, I would think that that would have a huge repercussion in terms of potentially moving forward. And it's like you're almost stopping a stone that you've started to get rolling after pushing it up a hill. And now it's like you almost have to push it back up the hill, right, to get the momentum back.
Yeah, that's exactly correct. That's not a naive way to look at it at all. I think, unfortunately, a lot of the things that we were hearing about sort of stopping things halfway is that there's sort of this idea of like, well, then you have half the data.
Mhm.
But the truth is, we talked about sample size, and we talked about making sure trials are exactly powered ethically to get the right answer, which means that if you were supposed to enroll 470 people in a study and you only had 100 people, that doesn't mean that you saved 2/3 of your money. Or I know that that's not the exact math of that proportion. But that doesn't mean that you saved 2/3 of your money. What it means is that you wasted 1/3 of your money, because it means that, and it also is extremely unethical, because it means that all of the contributions of the people who participated in that study, if you were to analyze their data, you may not actually get an answer because your sample size is too small to see a difference. And so your conclusion from the study is going to say something like, we did not see a difference between group A and group B, but the study was underpowered to find a difference. And so we therefore conclude that we need to do a bigger study. So what that means is that you're actually redoing all of your work again. And all of these people for whom we have, again, I think informed consent, participating in a clinical trial, I treat that as a sacred ethical contract with people. we are contractually, ethically obligated to see people through the end of the study. And even if we make sure that each individual person is safely through the trial, we're obligated to finish that trial so that we can then use their data for science to advance other people's care and come up with an answer for all of the time and blood and potentially risk that they took on to participate.
And the unfortunate thing, it sounds like, is you might not actually get that. Right, you take it through to the end, but it might not be statistically significant because of the precursor. And then my imagining is when you redo the experiment, you don't just lump in their data, right, and then get 2/3 of the new people. You have to start with a brand new cohort because of the importance of doing everything, in parallel.
Yeah, I mean, it depends. I think if it's something that was just paused, so for example, for a lot of our studies this summer, enrollment was paused, and which means that we followed people through to the end that were already on study and we didn't include more people.
Mhm.
If you, in relatively short order, reopen the study to enrollment again, then you can lump everybody together. But if you completely wind down a protocol, close it down, and analyze it, then it requires restarting a whole new study. And that's not just reopening it. It's probably writing a new protocol. It's getting ethics approval. It's getting FDA approval. It's kind of going all the way back to the drawing board. And so I really think that this idea of saving half your money turns into it's actually wasting half your money. And more than that, because really, before you even start enrolling people in a clinical trial, somebody's probably been at work at that for 5 years, if not longer. And that somebody is probably a whole team of people. And so, the cost of actually doing the clinical trial, of actually enrolling the people, is really a small component of that entire kind of stream of work that's been going on for years before that.
And when you're doing that restart, that whole precursor component too, like that 4 years, also you have to hit the reset button on. So it's not as simple as just like, we open back up the doors and let people back in. There's a lot of this backside work that also goes into it, and that's going to make it all the more difficult to, like you said, you're charging double or triple the amount that you're originally going for. So these donations, while small, y'all, can help maintain, right, the studies, the clinical trials, the people maintained in them, and hopefully that you could only at worst have that pause like you were mentioning during the summer, and then instead of like just a whole wind down, which is wild to think about. It would be like you said, almost the ethical obligation that you have to the patients. The wind down is unfortunate. Yeah, like what's the best way to help and advocate for something like this for a person at home? Like there's donations, and what would be some way that other people could get involved as well?
I think talk to your congressional representatives. I think in past years, science and politics were a lot less intertwined. And I think that regardless of who's representing you in Congress, I think it's really important to just let them know how important science and public health are to you. You can advocate in a way that's completely nonpartisan. You can just talk about how important it is to maintain our public health infrastructure, to maintain the CDC, to maintain funding to the NIH, to maintain funding to research institutes like the Fred Hutch, because it really is, again, all of this infrastructure that we've built helps the whole world, not just the United States. But I think that it's really important that they hear that, and that they hear from people, not just people impacted by HIV, but people with autoimmune disease, with cancer, with people with neurologic problems, developmental diseases, with dementia, because science really cuts across all of those conditions. And we can't really say that we're just going to like stop funding one part of science, or that we're going to stop paying attention to one part of health, because obviously our bodies, our minds are connected as an individual, but as a global community, we're also all connected. And so I think reinstilling that faith in science, the trust in science, making sure that the folks who are making these decisions for us are understanding how important science and public health are to every one of us, regardless of what we believe, what kind of medical conditions we have. We all need science.
Yeah, I love that. I love that. So one thing that you had mentioned was the importance of public buy-in to this, right? Of like, regardless
Mhm.
of what you're doing in terms of any kind of clinical trial, whether it be on HIV, AIDS, cancer, anything else, if you don't have public buy-in, it's not going to work. Can you touch a little bit about how, we mentioned that there's on the advisory boards, there's your everyday persons on those as well and being pulled in. What are some other components that you feel work and are helpful in terms of bringing more people on and like just getting that public buy-in?
I think just education, people knowing and speaking about it. I think if anybody comes away having learned one thing today, let that be that undetectable is untransmissible, meaning people whose HIV is treated cannot pass it on, have zero risk of passing it on. Drop it into conversation with somebody. I mean, that may sound really weird. I do it all the time. Then again, I had to ask my kid in the car today if they found my condom earring under the car seat. So,
Did they?
you know, uh, they did. And then I said, I'm sorry, that's kind of a weird question coming from mom. And my kid said, no, not from you. That's a totally normal question coming from you. But yeah, I mean, just drop it into conversation. Just, if people are saying things that don't make sense, or you're having a random conversation, just ask people, hey, did you know that people who take HIV treatment can't pass it on to anybody else, that there's zero risk of acquiring HIV? Hey, did you know that with 2 shots a year, there is zero risk of acquiring HIV, but that medication costs as much as a mid-sized sedan? And so we need to work on ways to, make that more affordable for people globally to get. Just get the education out there, which is what you're doing. And I so appreciate you for that.
And it's like furthering the destigmatization, right? Because you mentioned too, there is like the associated stigma of even getting that daily pill, right? And even if it's not out in the world in front of everyone, just like your own mental perception of it, right? And so
Yep.
it's really cool that you're not just a doctor of health, right? You're almost a doctor of the spirit too, that you have to take into account for these kinds of things. And I just, I think that makes the work that you're doing all the more incredible because it is so important. And I love to see that it's important to you, the scientist who's helping the people, right? Beyond just here's a drug, take this, right? And it kind of
Oh,
goes that like
absolutely.
really amazing like bedside manner that we have to like ensure that it's helping people to the max.
Absolutely. I mean, I have to say, as someone who takes care of lots of folks with HIV, sometimes when folks come in to see me, we don't even talk about HIV. Depending on if I even need to, I may or may not ask them how well they're taking their medication, because it may be obvious that they're doing great. And we talk about every other thing under the sun: mental health, substance use, aging, all the stress that folks have in their life. Like that's what keeps me going, is, getting to share that with people, sharing stories, being able to witness lives. Because the truth is that on a one-to-one personal level, folks who are accessing medical care, HIV treatment is so easy. It's fixing the rest of the system that's preventing everybody from being able to participate in that, being able to, get their head around this condition that we've built so much stigma into as a society, really for no reason.
Yeah, well said. What is something that you are most excited about that's upcoming in your research? Like, is there an experiment or an upcoming drug? I know you can't necessarily tell us about like the fine details of it, but just something where you're like, oh man, I'm like really excited for this. This is gonna be so cool, or it has the potential to be so cool, or right, like any of that, whether it be on the HIV side of things or a side drug. What are you most excited about?
Um, I think it's a couple of the upcoming studies that we have for long acting treatment for HIV. So we have a study coming up that will open just in the United States, that's using 2 different long acting HIV medications that will be given, one every 2 months, one every 6 months. And another study that's going to be using that same drug combination, and people will be randomized to take that long acting combination versus continuing a medication called TLD, which is a triple therapy drug, the generic drug that's available in most countries around the world. And both of those studies are for people for whom HIV treatment is failing, because they can't take a daily pill. Notice I said they're not failing treatment; the treatment is failing them. So I'm really excited about these studies, because oftentimes when we do research, we try to find like the exact perfect person for research. And these two studies are really saying, okay, who are the people who are not being served by current HIV treatment? And can we take these medications that we already know work well in people who take their medication easily and see if these treatments work for them, and figure out whether this dosing schedule and this way of giving it can really make an impact for people who are struggling to take their medication every day.
I love how you phrased it, and I love the excitement for it too. It's gonna be one of those big game changers, I feel, and just making it all the easier for, again, like you said, the accessibility, right? Then the treatment, the long-term treatment as well, and the injections, and possibly whatever else that's going to affect, right? Any other kinds of infectious treatments on that same long-term run. As we wind down, one question that we've asked all of our female scientists is, you know, we've kind of touched on this throughout, is that you're a badass woman in STEM, right? Is that you've got the kiddos, you got the family, you've got an incredible research program. Like, I hope everyone's jaw has been hitting the floor on everything that you've been talking about tonight, because it is that impressive. But can you just like highlight a little bit for people, what has it been like for you as a woman in STEM, and what advice would you give to any up and coming women who are watching, listening, maybe interested in science? How would they be able to really get into the sciences, and, just knowing what you know now, what would you tell them?
Gosh, I mean, I think it's always more difficult to be a woman in STEM than a man. I think something that I think about a lot as a white woman in STEM, so I'll just flip the question even more, is that a white woman in STEM, that I don't think that I face as many challenges as a lot of other underrepresented groups in STEM. Specifically in infectious disease, there are a large number of women. My division head's a woman in both my institutions. I have a lot of great female mentors, a lot of great female leaders, but there are a lot of people who are not well represented, Black and brown physician scientists, people belonging to other underrepresented communities, and folks who didn't come into science with sort of intergenerational wealth. And I think that's really probably the most challenging thing, is to get people who don't see themselves in science into science. And, you've talked about the fact that I think financially it's very challenging to be a scientist, and there's a kind of a big area of training in which people don't make a lot of money and work very hard. And for folks who don't have a partner who can support their family, or who come in with a lot of debt, or maybe they're the first person in their family in college, first gen students, like that chasm can be really, really difficult to cross. And so I think I'm trying to call in, I specifically am trying to call in my, other people who are already established in the field, to make sure that everybody is welcome. Everybody is welcome in STEM.
I love that. I love it. Well, thank you very much for your time tonight, for sharing your expertise with us and all of your amazing insights. I hope folks have enjoyed our discussion. I know, again, like, you're listening later on, or you're watching, my jaw was probably dropping for most of it. It was just, again, so much fun, and thank you for your time, and, again, sharing your knowledge with us. We appreciate you. And guys, again, you can support this incredible work by hitting that dono link in the chat right now, and supporting Dr. Bender Ignacio's work.
Thank you.
Thank you again.
Thank you so much for hosting me. I've had a great time speaking with you, and it's really generous of you to let us, use your platform to share more wonderful science and to talk about the work that we do. So I really appreciate you, having me on.
Likewise. Thank you so much. And we hope to get you back soon. If you ever want to come on and talk about any of your science, we'd love to have you back.
Absolutely. Great.
Thank you again. Have a good night. Everyone.
Science is for everyone, all backgrounds, all ages, all genders, and all walks of life deserve an education. Come be one of us.